Okay, so this is chapter 7, part 1, and in this chapter we're going to talk about cellular respiration. Before we jump into that process, I just want to remind you, we've seen this a little bit so far this semester in a couple of different places. I want to remind you of the two main metabolic pathways that we're going to study. In this chapter, we're going to be focusing on cellular respiration, and in the next chapter, we're going to be focusing on photosynthesis. And I want to show you how these two are tightly tied together. And so we'll start up here. You can see these yellow arrows. This is light energy coming from the sun. That'll be in the form of waves, right? That energy is going to be captured by photosynthetic organisms like plants. It gets captured by a very special organelle called the chloroplast. And inside the chloroplast, there's going to be the pigment molecule called chlorophyll, which again we'll talk about in detail in the next chapter. And that pigment molecule is responsible for the absorption of energy from the sun. Inside of this organelle, that energy can then be converted into chemical energy, so organic molecules. And in that process, oxygen is produced as a byproduct, which is really a waste product of photosynthesis. And then these organic molecules with oxygen are going to be broken down in a special compartment in eukaryotic organisms called the mitochondria. And so that's the organelle that we're going to be talking about really in this chapter. And so this process called cellular respiration, all organisms on the planet are going to do some form of this, either aerobic or anaerobic. Where organic molecules will be taken. In this case, if it's in the presence of oxygen, we would call that aerobic cellular respiration. So that term aerobic tells us oxygen is present. Okay, so cellular respiration happens inside the mitochondria. Organic molecules will be broken down and energy will be pulled out of those organic molecules as bonds are broken. And that energy is going to eventually form ATP, which is, remember again, the energy currency that our cells are capable of using to drive cellular work. Okay, some of that energy, of course, is going to be lost as heat, right? Because no energy transformation is 100% efficient. Photosynthesis is not 100% efficient, right? Not all of the energy coming from the sun is completely captured and completely turned into organic molecules. And the same thing, not all of the energy in organic molecules can be pulled out to make ATP. Some will be lost as heat. Remember that second law of thermodynamics. Okay. Other products of cellular respiration besides ATP, which is the one that we're most concerned about, but we also produce water and carbon dioxide as byproducts of cellular respiration. Okay. So again, this is just kind of an overview picture. Really connecting these two metabolic processes. Okay. So again, in this chapter, we're going to be focusing on cellular respiration, and in the next chapter, we'll focus on photosynthesis. And so before we get into the steps of cellular respiration, I want to just introduce you to what exactly it is, and then I want to tell you about an alternative pathway called fermentation. So both cellular respiration and fermentation are exergonic catabolic processes, right? And so you should automatically think back to chapter 6. Exergonic means reactions that release energy, right? The change in free energy value is negative, and therefore energy is released. And catabolic means breakdown. So we're going to take larger molecules and break them down. In aerobic cellular respiration, remember this is going to be a process that converts chemical energy stored in the bonds of organic molecules to ATP. When you see the term aerobic, I want you to think requires oxygen. And this product here, ATP, is going to be what cells can use directly to do work. We've already talked about processes that require energy in the cell. For example, active transport requires energy. Where does that energy come from? Cellular respiration. Building anabolic processes that require energy, endergonic reactions, Where does that energy come from? Cellular respiration. Okay. The alternative pathway here, fermentation, organisms that are anaerobic will do this process. And we'll talk about fermentation at the end of this chapter. I just wanted to give you a little bit of an idea of what it is. It's similar to cellular respiration. Again, it doesn't require oxygen. Really, it's just the first step of cellular respiration, which consists of 4 steps, which we're going to learn about. In the process of fermentation, we say that the fuel source, which is glucose, is not completely oxidized, which means that not all of the energy is pulled out of glucose. So it's a pretty inefficient process. It releases much less energy, and so less ATP is made by fermentation. But again, organisms that can't use oxygen, or if oxygen is toxic to them, they don't have the pathway of cellular respiration, and so they're kind of forced to do fermentation. So again, we'll come back to that at the end of the chapter. But for now, our focus will be on aerobic cellular respiration. Okay, so again, here is the formula. You're going to have to know this. So C6H12O6, this represents glucose, plus 6 molecules of O2. This is oxygen, or we could say molecular oxygen. And that's going to yield, here's our arrow, 6 molecules of carbon dioxide, 6 molecules of water, H2O, plus energy in the form of ATP. Okay, so remember, on the left side of our equation are our reactant molecules, and on the right side of this equation are our products. This process, this formula, we say is a redox reaction. And so again, remember back to chapter 6, we briefly talked about this. A redox reaction is where there is a reduction and an oxidation. Okay, I think I said the little mnemonic, Leo the lion goes grr. So the molecule that is losing electrons we say is oxidized, and the molecule that gains those electrons is reduced. Okay, so Leo, lose electrons, oxidized. The lion goes grr, gain electrons, reduced. That's a little way to remember it. Okay, so in this equation, it's glucose that's going to lose electrons, and we say that glucose is oxidized. And as it loses those electrons, it loses energy. Because remember, electrons represent stored potential energy. Okay, we say that oxygen, the other reactant molecule, which will eventually gain those electrons, becomes reduced. So glucose is oxidized, oxygen is reduced. Okay, now glucose doesn't just lose its electrons and oxygen gain them right away. It's not just one step. Instead, we're going to see that it's a series of steps. So electrons are slowly pulled off of glucose. They're not given directly to oxygen. First, they're given to this molecule right here, NAD, which becomes NADH. We learned about this back in chapter 6. When you hear NADH or FADH2, which you're going to hear in this chapter as well, both of those molecules I want you to think of as electron shuttlers. Their job is to transport electrons from where they're being pulled, right, to the very last step in this process, which is called the electron transport chain. And we won't talk about the electron transport chain until part 3 of this chapter. Okay, so right now you just have to realize electrons are going to be pulled off of glucose. As they're being pulled off, they go to NAD to form NADH. And NADH is going to eventually bring those electrons to the electron transport chain, where they finally, after they lose all of their energy, are given to oxygen to form water. Okay, so again, it's not just pull electrons off here, give it to oxygen. There's this series of steps, these slow series of steps. So the electrons lose a little bit of energy along the way. That's how our cells are able to harness the energy from glucose. And again, the process is still not 100% efficient. So let's look at a quick overview before we get into the nitty-gritty here. And so these are the 4 steps that we're going to talk about. Okay, some books will do this in 3 steps, And sometimes we do it in 4 steps, depending on what book you're reading, depending on who your instructor is. The book that we are using breaks it down into 4 steps. And so that's what this figure shows. So that's how we're going to do it. So the first step is going to be called glycolysis, and we'll talk about glycolysis right in this first part of the chapter. Glycolysis, if we split the word up, glycolysis, it's literally the splitting of glucose. And so the starting molecule for glycolysis is glucose. It's a 6-carbon sugar. It gets split into 2 3-carbon molecules that are called pyruvate. So pyruvate, 2 of them, are the end product of glycolysis. Now, a little bit of ATP is made here. We'll talk about on the next slide how. That pyruvate, both molecules, are then moved into the mitochondria, and everything from here over happens inside of the mitochondria. Glycolysis happens in the cytoplasm. So the second step, so pyruvate enters the mitochondria, and we've got some different regions of the mitochondria, and there's actually a labeled figure on the next slide, but just to give you an idea, the very center region is called the matrix of the mitochondria. We then have an outer and an inner membrane. So I'm pointing here. Here's the outer membrane, and here's the inner membrane. Let me just highlight those. So, okay, so outer membrane here, surrounding the entire mitochondria. Inner membrane here. Notice the inner membrane's folds. These are called cristae. We're going to come back to these when we talk about the electron transport chain. The very center region here is filled with a fluid. It's called the matrix. And then this space in between these 2 membranes is called the inner membrane space. So all of these components here, all these parts and pieces to the mitochondria are really important. So you want to make sure you can draw and label a mitochondria. Okay. Get rid of that. So pyruvate enters the matrix of the mitochondria, and that's where the second step occurs, which is called the formation of acetyl-CoA. We'll talk about this in the second part of the chapter. Acetyl-CoA, 2 of them are formed, and both of them move into the citric acid cycle. And by the end of the citric acid cycle, we say that glucose has been completely oxidized. And again, a little bit of ATP is made here too. And then all of the NADH and FADH2 that's made during glycolysis, the formation of acetyl-CoA and the citric acid cycle, all of those molecules end up here at the electron transport chain. And this process, again, we won't talk about until the third part of this chapter. It happens on the intermembrane. This is where the membrane proteins are located. And this is where the bulk of the energy is made. So about 32-ish ATP are made during the electron transport chain in chemiosmosis. Okay, now the ATP made during the citric acid cycle and the ATP made during glycolysis, there's just a little bit. It's made by something called substrate level phosphorylation, where the ATP made here at the end is made by chemiosmosis. And so I just want to briefly talk about the difference. Okay. So we'll talk about chemiosmosis first. Here's a nice labeled diagram. So again, you should be able to recreate this. You should know all the parts and pieces of that mitochondria. So again, most of the ATP, the bulk of it, okay, if we're talking about 36 ATP being made, 32 of those are made by this process called chemiosmosis and what we call oxidative phosphorylation, which again is the third part of the chapter. So we're going to really talk about that. This takes place in the mitochondria. Specifically, the proteins involved in this process are located on the inner mitochondrial membrane. Okay, the intermembrane space is also involved here. And again, we'll talk more about that at the end. Again, the small amount of ATP made during glycolysis in the citric acid cycle is made differently. It's made by something called substrate level phosphorylation. And this is a pretty easy process to understand. What happens is a phosphate group is taken off of some intermediate reactant molecule, and it's given or transferred to adenosine diphosphate, ADP, to make ATP, adenosine triphosphate. So literally an enzyme just comes along and it just snatches a phosphate group from an intermediate molecule and gives it to ADP. Okay, so pretty easy to understand that. Okay, so we're going to end this part of the chapter off by just talking about that first stage, glycolysis. Okay, and we'll go over the specifics. So glycolysis is the first step in cellular respiration. Again, this process takes place in the cytoplasm of the cell. Everything else happens in the mitochondria. This is the only step that happens in the cytoplasm. Importantly, I want to just skip to this point, number 4. This process does not require oxygen. Okay, so glycolysis can occur in the presence or absence of oxygen. This first part will run. Okay. Overall, what happens is glucose is our starting material. It's our reactant. It's a 6-carbon sugar, right? It gets split into 2 3-carbon molecules called pyruvate. So in the end, we get 2 molecules of pyruvate. There are 2 phases to glycolysis. We have what is called the energy investment phase, and that's going to be followed by the energy payoff phase. And so what we have to do is we got to put a little bit of ATP in to get the products out, which we end up with more ATP. So overall, the process is exergonic. It has a negative change in G value. We also get NADH out of this process. So again, the NADH produced is important, right? Because that's going to go on to the electron transport chain where a lot of energy gets made. Okay. Now the pyruvate, the end product, goes on. It goes into the mitochondria and it gets further oxidized in there. So cellular respiration isn't over yet, right? This is just the first phase. So let's look at this. Let me move my camera. And so here it is in a nutshell. So at the very top, we have what's called the energy investment phase. So again, we're in the cytoplasm. We've got glucose. It's a 6-carbon sugar. We have to put 2 ATP in. So you can see 2 ATP used. Okay, and then we have the energy payoff phase. So what happens is we get out 4 ATP, we get out 2 NADH, and we end up with 2 molecules of pyruvate, which again are 3-carbon molecules. So here's the net. So we put glucose in, we get 2 pyruvate out. We put 2 ATP in, but we formed 4, so we have a net yield of 2 ATP. And then we end up with 2 NADH coming out. Okay, so that's the overall. I'll quickly show you the nitty-gritty, and I'll tell you that you're not responsible for knowing the structures or even really knowing the names of these molecules yet. If you are a biology major and you go on to take organic chemistry or biochemistry, I should say, you will probably have to eventually know the names of these. But for now, I'm not going to hold you to it. I'm just going to show you. So this is the energy investment phase on this slide. So here we start with glucose. That should look familiar. It's a 6-carbon sugar. Glucose is going to be phosphorylated to form glucose 6-phosphate. So on carbon number 6, there's a phosphate group. Okay. This process here is going to be catalyzed by this enzyme, which is called a hexokinase. And that's the first enzyme in this pathway. Remember that enzyme. What happens next is this undergoes a rearrangement. And so this enzyme here called phosphoglucoisomerase catalyzes that reaction where we change the shape of this molecule. So it becomes fructose 6-phosphate. So it just looks differently. It's got a different arrangement. It's an isomer. And then phosphofructokinase, this enzyme, is going to, again, phosphorylate. So ATP comes in and it dumps off a phosphate. We end up with fructose 1,6-bisphosphate. So carbon number 1 and carbon number 6 are phosphorylated. An enzyme here called an aldolase then splits this molecule, and we end up with glyceraldehyde 3-phosphate, or G3P. And we also end up with dihydroxyacetone phosphate. And these, again, are isomers of each other. And an enzyme called isomerase can actually switch these two back and forth. It's glyceraldehyde phosphate, also known as G3P, that enters the next phase. Okay? The next phase of this pathway is the payoff. And so, again, G3P, the product of the investment phase, enters. This enzyme, triose phosphate dehydrogenase, comes into play. It's going to strip off electrons and protons. And we end up forming NADH, okay, 2 of them, because we have 2 glyceraldehyde phosphates that are going to be coming in. This then becomes 1,3-bisphosphoglycerate. The enzyme phosphofructokinase then comes in, pulls off a phosphate from each of these. Remember, there's 2. We end up with 2 ATP being pulled out. We end up with 3 phosphoglycerate. Again, the enzyme phosphoglyceromutase comes in, changes the arrangement. So we have 2 phosphoglycerate. An enzyme called enolase pulls out water, and we end up with PEP, which is short for phosphoenolpyruvate. This then again gets the phosphate taken off. We end up forming 2 more ATP, and pyruvate kinase helps us to form pyruvate. Okay, so again, we put 2 ATP in, but on this slide you can see we're getting 4 out. So there's a net yield of 2 ATP. We also form 2 NADH molecules, which remember is reducing power. That's carrying electrons to the electron transport chain. And again, in the end, we end up with 2 molecules of pyruvate, okay, for each glucose. I just want to go back really quickly to this slide. So if you remember from chapter 6, we talked about feedback inhibition. And so this metabolic pathway is also under tight regulation, right? And so if the amount of ATP in the cell starts to build up, and the cell's not using it, the cell's not going to waste energy, time, on running this process. So if there's a buildup of ATP, we want to turn this off. And so what will happen, the best thing that's going to happen, is an early enzyme in the pathway will get turned off. So right here at this first step, so this enzyme hexokinase, if ATP builds up, this hexokinase will be turned off. ATP will bind to an allosteric site and actually render that enzyme inactive. If ATP levels are dropping, it comes off and the pathway runs. So it's a reversible reaction. Okay. All right, I want to take you back here. So we just did glycolysis. That's it. Okay. Net yield of 2 ATP right here. We also made 2 NADHs, which will eventually go into the mitochondria and be used here. The last step, which we're going to talk about a little later. We also end up with 2 molecules of pyruvate that also enters the mitochondria. And that's what's going to enter this second pathway, which is the formation of acetyl-CoA. So pyruvate still has energy in it, both molecules, right? And so those 2 molecules need to be further oxidized. So energy needs to continue to be pulled off or out of pyruvate. And that's what's going to happen in the next 2 steps. Okay? Okay. So part 2 of the chapter, which is coming up, we're going to look at step 2 and step 3. And then in part 3 of the chapter, we'll look at the electron transport chain and oxidative phosphorylation. And then we'll end the chapter again with anaerobic respiration fermentation.